So I'm Alexander Bershadsky, and at the moment I'm working in two places. One is my real affiliation, Weizmann Institute, and now I also have a laboratory at Mechanobiology Institute at the National University of Singapore. And we'll return to actin. Actually, all this information already somehow was kind of discussed here, but what is important? Actin is a protein which is most abundant protein of our body and which has a wonderful ability to form filaments. Now, what we should know about these filaments? These filaments, A, directional, so two ends are non-equal, and therefore, first of all, the assembly at two ends will be different, but also molecular motors can move directionally along these filaments. Second, and it will be important in my talk, this is helical structure. You can consider it as two right-hand helices, and the parameters of these helices is very kind of constant for all filaments made of actin. And the third important statement about actin, which will be necessary for us, is that in its assembly there are essentially two stages: nucleation and elongation. And nucleation is kinetically limiting stage because cell on purpose keeps the concentration of depolymerized actin very low using different kind of actin sequestering proteins. So spontaneous nucleation is proceeding very, very slowly. And this is on purpose because this is a nice place to regulate it. The cell has several types of protein devices which facilitate the nucleation, and this is a main tool to assemble actin in a proper place in a proper time. Now, actin was discovered in 1942 essentially as a muscle protein. Actually, a laboratory of St. George studied muscle contraction. But the real breakthrough appeared in 70s when Elias Lazaridis and Klaus Weber succeeded to obtain antibody to actin, and then when they stained cells, just fibroblasts in the culture, they found these very nice bundles or fibers, and this was found in many cell types, which suggest that not only in the muscle but in all our cells you have a lot of actin, and this actin is really organized into very ordered structures. They of course very ordered even in this not very perfect image, but already at that time they noticed that a multitude of patterns have been observed with varying degrees of complexity, and each cell appears to have its own individual way of portraying its actin filamentous network. So until now it is not very clear how really cell form ordered array of these actin bundles, and this will be the main subject of my talk today. Now we know that a very important feature of these actin bundles, each of which consists of 100 or if not 1,000 individual actin filaments, is their association with integrin-mediated cell adhesions with the extracellular matrix shown here in yellow. And these adhesions are also actin-based structures which have a unique feature to sense the force that applied to them. So actually, this is I will not discuss in detail, but it is relatively well established that these focal adhesions are growing upon application of force from the actin cytoskeleton, and moreover, they can nucleate actin bundles themselves, and the growth of actin from these focal adhesions is also force dependent. The first association with, of these stress fibers with adhesions was found by Graham Dunn, who is still alive, and I met him in London recently. So since 1978 we know that, but we still do not know how the whole system is arranged. Of course, besides these bundles, there are many other actin structures that already were mentioned here. There is this actin cortex underneath of the cell membrane that Eva discussed. There are lamellipodia, which contain the branching network of actin. There are filopodia that contain the bundle of parallel actin filaments. And also there is a network that filling the whole cytoplasm. So what I'm going to do is first, for pre-warming, to discuss here how this general network filling the whole cytoplasm is organized. And then the major part of this talk will be how these bundles of actin filaments and focal adhesions emerge and how some order appeared. And finally, I will show you some new data on organization of myosin that we obtained using so-called structural illumination microscopy, which kind of open a new facet in organization of the cytoskeleton. Now, first I should make a very brief review of numerous proteins that orchestrate the assembly of all these structures. There is at least 60 classes of actin binding proteins, which means that it is about 500 types of individual proteins. Among them, of course, many different type of cross-linkers. There are proteins that regulate assembly by kind of blocking it at one or another end of actin filaments. There are, as I already mentioned, monomer sequestering protein. There are proteins that cut actin filament into pieces, but most important and what I will kind of concentrate today are the proteins that facilitate nucleation. And second class of protein are myosin type motors that are moving along the actin filament. Now, about nucleation, there are actually two devices that permit to kind of facilitate nucleation of actin filament. One is ARP2/3 complex, which already was mentioned here, and which consists of actin-related protein 2 and actin-related protein 3, which are close, really close relatives of actin, very similar to it, plus 5 other accessory proteins. When this complex binds to the site of pre-existing actin filament, and then another actin molecule bind here, you have an imitation of nucleus consisting of 3 monomers, and this nucleus can then give rise to the newly formed polymerizing actin filament. Of course, this way of nucleation permit only formation of branching network of actin, which is very useful when you need to push something, for example, membrane forward. However, for many other purposes, you should nucleate linear filament, non-branching filament, and this is mediated by another class of proteins that was less frequently mentioned here, and these proteins called formins. Formins is a family of proteins that can form dimer. There is a special formin homology domain 2 which dimerize in ring-shaped structure, and this ring can kind of gather 3 subunits of actin which facilitate the nucleation. But in addition to that, when actin filament already existing, this dimer of formin can bind to or can remain at the growing end of actin filament, but permit incorporation of new actin monomers. And if these actin monomers are associated with a protein called profilin, which is a major protein that's sequestering actin, profilin has affinity to another domain of formin called formin homology domain 1, and therefore efficient concentration of actin in the proximity of growing end of the filament becoming high. So formins strongly promote also the process of elongation of actin filaments. Formins then not only nucleators but also elongators of actin filament, and typical structure that mediated by formins is philopodia, where formins are sitting at the tip and promote formation of the bundle of actin filament, which is a core of this finger-like projection. Unlike ARP2-3 complex, which is single, you have several genes encoding this FH1 and FH2 domain, and mammal you have 15 genes of this type, and we know about function on only few of them, but definitely they play a major role in organization of the actin cytoskeleton. And the last textbook slide is about myosin. There is 24 classes of myosin type proteins. Among them, all are moving along actin filament from minus to plus end, and only one, myosin 6, is moving in opposite direction. And there are of course many ways to use these different myosins for moving of actin filaments, but only one type of myosin called myosin 2 has long tail domain that permit formation of bipolar filaments, and this device permit the movement of oppositely oriented actin filaments towards each other, which is the basis of contraction. And particularly the muscles are consisting of repeating units of this kind called sarcomeres, and upon interactions of myosin bipolar filaments with 2 arrays of actin, muscle contracting, and this is of course very specialized but common way to induce contractility in all cell types. Now I'm going to discuss first part of my talk, namely organization of the actin cytoskeleton in local in the cytoplasm. And the motivation for that is the observation that made several years ago in Mike Sheets' laboratory, that if you inhibit myosin in the cell, you not only block contractility, you actually make the whole cytoplasm of the cell fragile and, as he called it, non-coherent. For example, this is control cells which is spreading, and you see typical discoid cells, but if such cell is spreading in the presence of inhibitor of myosin, you see that here and there the big holes in the cytoplasm can be formed. So it appears that the cytoplasm cannot resist anymore the membrane tension, And yeah.
You're actually showing the ROC inhibitor, so there are other pathways downstream of ROC that is affected. Right? So
Yes, but in this case it is inhibition of ROC kinase, which is known to be the major regulator of myosin 2 in the cell, but this can be reproduced using the absolutely specific inhibitor of myosin. So there is something in the cytoplasm which keep it together, which depends on myosin 2. So we decided together with Mike Schitz to look more precisely on the organization of this network that filling the whole cytoplasm. And for that we used a simple setup. We plated cells on these narrow adhesive strips so that they form bridges above this strip. So we concentrated on the region of the cell which are not related to adhesion, just form this bridge above the non-adhesive substrate and try to look what is the organization of the cytoplasm in this cell. The cells were labeled simultaneously with a marker of actin and myosin light chain. And what we have noticed using some trick, namely we treat these cells with a small doses of latrunculin A, which does not change essentially the cell shape and even do not prevent actually actin polymerization, but by some reason making much more clear what is going on in the cell. And what you see here is formation of numerous patches that you will see now. And these patches are moving in the cell quite vigorously. At first, the actin and myosin do not coincide, but if you wait a little bit more, you will see that the more you are waiting, the more is colocalization of actin and myosin. We can measure this movement. For example, we can look on this picture and put it, this is time axis and these are just seconds, and you see this three-dimensional chemogram showing clearly how these patches are moving, both for actin and myosin. You see that in the beginning they do not coincide, in the end they becoming more colocalized, and probably it is not surprising. And we can of course measure their movement simply by measurement of mean square displacement, and it is not surprising that any methods of inhibition of myosin, either by knockdown of myosin 2A or by treatment with blebistatin, which is a specific inhibitor of myosin 2, you can significantly suppress this movement of these patches. Here you have this chemogram before blebistatin, and after blebistatin everything is essentially frozen. Interesting that in addition to actin and myosin, there is a formin, which I mentioned already. This formin called DAM1, one of the members of this formin family, which can be found in these patches. Again, we visualize it with treatment with latrunculin, and you see numerous patches which contain both actin and DAM1 formin. And in a way, this is a proof that such patches are not created by latrunculin treatment, because if you look on non-treated cells, you will see that this, using palm super resolution microscopy, you will see that patches of this formin DAM1 can be easily found in the cells that was not treated with latrunculin. And what is interesting, that inhibition of this formin, either by knockdown or essentially by knockdown, or we use low molecular weight forming inhibitor, about which I will speak a little bit later, also block the movement of these patches. The third protein that we found there was one of the actin cross-linking protein, filamin A, and I will not repeat kind of the same images, but altogether it seems that the structure of the network inside the cell that filling the whole cytoplasm can be described as this aster-like structure with forming inside and plus ends of actin filaments close to the center, and myosin in between. Of course, myosin dimers in between. And Alex Magilner, together with Jean Lard, made a simulation of such structure. And red here is a myosin, and you can kind of see how these processes of fusion of